What is epistasis? Make a note that epistasis actually is not covered in EdXLB, so don't worry about it. Epistasis basically describes a situation where the expression of one gene can affect the expression of another totally different gene on a totally different chromosome, or rather it can mask the expression of a phenotype from a particular gene. These genes basically are said to act either antagonistically or in a complementary fashion. What does it mean? Either the expression of one gene masks the expression of the other, or the expression of one gene is required to facilitate the expression of the other. That's what complementary means. Recessive epistasis occurs when we have a homozygous recessive genotype, and it will block the expression of another gene because normally that gene was required to facilitate the expression of the other gene. On the other hand, a dominant epistasis occurs when we have a dominant genotype that blocks the expression of another gene. So this concerns genes that act antagonistically. This concerns genes that act in a complementary fashion. Let's see this little example here of, for example, and whenever you have epistasis questions, how will you know? You'll know because you're going to be given some kind of context like that. You're going to be given some sort of signaling cascade, or like a cascade of enzymes and proteins working together. So, for example, these are some precursor proteins to produce this pigment here, anthocyanin, on plants. It exists as a precursor, which is by an enzyme that is coded for by a gene, is converted to another precursor. You don't need to know the intermediate names. Yet another gene that codes for another enzyme converts it into a third precursor. Yet another gene converts it into its final form, which is anthocyanin. So let's, for example, say that gene A determines if a banding pattern is present in mice. Allele A leads to a banding pattern, and a lowercase a leads to a pure black color. A second gene, B, determines whether melanin is going to be synthesized at all, because without melanin, we can't have a black color, and we can't even have a banding color because there's no pigment there. Allele upper B leads to the production of melanin, lowercase b prevents any color formation. So it's a mutated version of the gene that codes for melanin. That doesn't allow the animal to make any melanin. A homozygous dominant and a recessive individual were crossed. Their offspring were subsequently crossed after. So if they were both homozygous, one for the dominant and one for the recessive, their offspring would all have been heterozygous. And it is the offspring that we cross together, which would theoretically lead to a classic, if it was a normal, or a usual rather, dihybrid inheritance with no epistasis, no linkage, no sex linkage happening, then we would theoretically be expecting a 9:3:1 ratio, wouldn't we? However, here we are going to notice a deviation from that genotype, the phenotype ratios. If we were to perform a chi-squared test, we would probably find a significant result, and it would tell us that there's some kind of underlying biological reason why we're observing the deviation, and one of those reasons can be epistasis, as can be sex linkage or codominance. Okay. Let's go ahead and write out the gametes and the phenotypes. So calculate, coming up with the different types of gametes that I can have. I can have this, sorry, it's meant to be a B. I can have this and this. Same for the other parent. So if I now go ahead and fill out my Punnett square, I'm going to try and do this a little bit quickly because we've run over a little bit. We ran over from the previous session. So because of that technical little glitch here, which I apologize for, but our day is going to finish just like about 15 minutes later, so hopefully that's not going to be a problem for you guys. Because I don't want to eat our breaks. It's impossible to do this without any breaks. So the way that you do the Punnett square is pretty much the same as when you have a classic dihybrid cross, right? It's going to be at the stage of, at the stage of interpreting the phenotypes from the genotypes that we're going to get a different ratio, as you're going to see in 2 seconds. And do feel free to write this along with me. I'm not entirely sure whether this particular set of answers is going to be set out to you. Right. Let's go ahead and calculate the phenotypes now. So how do we do that? If we have A, so what are we told? We are told that the lowercase allele B prevents any melanin production, basically. So anywhere where I see this bit of phenotype, I know that I'm going to have a completely white animal because there's no melanin produced. I can't speak about white or banded. If I have someone that has allele A, then they will be banded. So this individual here, let's just change color. Let's do that. So this individual here is A, so they're banded. And because they also they have the dominant gene for creating melanin. So they'll be banded. Banded, 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 and banded, and banded. And also here, banded. Okay. We are also, though, going to have, let's change color. Let's go for green. This individual is going to be white because no melanin. This will be white, and this will be white, and this will be white because there's no melanin. Finally, sorry, I missed one banded. Here, banded. Finally, what is our final? Let me just do purple. What is our final genotype? This will be black because they've got this allele that leads to black. So black, black, but they do have melanin, black. So what is my actual then observed phenotypes? 9, which are the banded ones, 2, 4, which are the white ones, 1, 2, 3, 4, 2, 3, which are the black ones. So a deviation from the 9331 genotype. Let's do a final question. Alfalfa seed color is determined by 2 genes which interact in the following way. Precursor 1 is colorless, 2 is orange, and xanthophyll is yellow. Precursor 1, so this is the diagram. State the phenotypes of the following genotypes. I'm just going to give you answers real quick because we've really overrun. So this one is going to be yellow because xanthophyll is produced. This one is going to be colorless because precursor 1 wasn't able to be changed by enzyme A. And this one here is going to be orange because precursor 2 wasn't able to be converted to xanthophyll, which is yellow.